Hello everyone, welcome to MMBS111 Introductory Microbiology. This is mini lecture 11.1 where we will be discussing interactions between microbes and their human host. There are 4 learning outcomes for this mini lecture. First, differentiate between the terms colonization, infection, and disease. Second, where would we find the microbiota or natural biota on and in humans? Third, when and how do we acquire our microbiota? And fourth, what is the Human Microbiome Project and what are we learning from it? So hopefully you have picked up by now in our earlier chapter discussions that the human body is colonized by a number of different organisms, and sometimes those organisms cause disease, but many times they do not. And our body is in a constant state of sort of dynamic equilibrium with microorganisms, and that equilibrium is considered dynamic because it's always changing. It doesn't remain in the same state from birth to death. The organisms change, numbers change, and typically an individual who is healthy maintains a very peaceful and mutualistic coexistence with those microbes. Occasionally, though, the balance can tip in favor of the microbes, and then disease can result. So some terms that you need to be familiar with are colonization. And when we say colonization, we're talking about microorganisms that are growing on body surfaces. However, those microbes are not causing disease. So they're simply colonizers looking for a place to live. The microbiota is the collection of all the microorganisms living in association with the human body. The microbiome is the collection of all the genes of the microbiota. So when we say microbiota, we're talking about organisms. When we say microbiome, we're talking about genetic material. And infection is kind of the opposite of colonization. Infection happens when tissues are invaded by microbes, and that results in disease. Okay. The Human Microbiome Project is a project that was started in 2008, and it was a 5-year project. And the goal of the project was to do a number of things, to characterize the microbes that were living on healthy individuals, and then to make some comparisons and determine how the microbiome in healthy individuals differed from individuals who were experiencing a variety of different diseases. The Human Microbiome Project utilized a number of really innovative and powerful techniques to sequence genes within that microbiome. And we discovered that human cells possess 22,000 protein encoding genes. So the 22,000 genes that are responsible for the numerous proteins that our cells produce. The microbiome, so the genetics of all of the microorganisms living on the human body, resulted in 8 million protein encoding genes. And these genes, interestingly, have an impact on our cellular genes and the expression of certain genes. And we're still learning a lot of things about the relationship between the microorganisms that colonize our bodies and their relationship between disease and our health. The image there on the right really shows you kind of how the microbiome can vary and change. And there are a number of variables that contribute to our microbiome, and it can be related to our lifestyle, so how we live, what we eat, activity, inactivity, the environment that we live in. So scientists have discovered that there are some differences in the microbiome depending on geography and where you live, the condition of our immune system, our contact with other members of the community. There are a lot of things that contribute and change our microbiome. So what have we learned from the Human Microbiome Project? Well, a number of things, and we're still learning from this project. We've really begun to understand that microorganisms are essential to the function of our cells. They are involved in metabolism of indigestible carbohydrates. They produce vitamins and hormones. They are essential to the proper development of our immune system and maintaining homeostasis within that system. And they provide some essential protection on those mucous membranes. When we experience dysbiosis in our microbiome, that generally is associated with disease. And the image here shows you the relationship, or I guess not really relationship, but it shows you how our microbiome can be altered or changed depending on disease. And we're still really learning some of these things, but there is a lot of research done and a lot of information out there that really supports the idea of this relationship between a healthy microbiome and one that is not healthy and contributes to a number of different diseases. So how do we get our microbiota, our normal flora that exists on our bodies? Most areas of the body that are in contact with the outside environment will be colonized by microorganisms, those resident organisms that live our entire lives on those surfaces. Typically, internal organs and tissue and those fluids are microbe-free. The microbes that colonize our bodies have really developed a very unique and complex relationship with us and our cells. This image on the slide is from your text, and it illustrates how our understanding of the microbiota has changed with the Human Genome Project, and how our understanding has developed. So it was believed for a very long time that lungs, the bladder and urine, breast milk, and amniotic fluid in the fetus were sterile, that there were no organisms that colonized those fluids or organs. We have since discovered that that is not the case, and there are a number of organisms that colonize all of these structures and fluids, including urine. We have also discovered DNA from microbes in the brain and the bloodstream. We have not isolated live organisms, though, so we're still trying to understand how that DNA has entered those locations and what its purpose is. So on our body surfaces and structures, there is this antagonistic relationship going on between our normal flora or microbiota and those organisms that are considered pathogenic or opportunistic. So in a healthy individual, the normal biota is unlikely to be displaced by new microbes. And the reason for this is that there are limited attachment sites on and in the body for new organisms. Another reason is that our normal flora creates a chemical and physiological environment that is hostile to other microbes. So our normal flora does a very good job of protecting us from invading organisms. They also help to keep numbers in check. So it's possible for disease to occur if numbers of certain organisms grow out of control. And our normal flora tries to really regulate the numbers of organisms present and the types of organisms. There are a number of things, though, that can weaken those defenses and make us more susceptible to infection. The first is age. The old or elderly and the extreme young generally have weaker host defenses. They have fewer organisms that naturally colonize their bodies, and their immune system has not been fully trained by those organisms yet. As we age, our normal flora really changes. Our immune system also ages, so we become more susceptible to certain infections. Genetic defects and acquired defects in our immune system can also increase our susceptibility and weaken our host defenses. Anytime we undergo surgery, and particularly organ transplants, because we generally are put on an immune suppressive drug, that can weaken your immune system and make you more susceptible. Any other underlying diseases like diabetes, cancer, liver malfunction, any of those kinds of things can also make you more susceptible to an infection. Stress also can increase your susceptibility to an infection. Pregnancy also, because the immune system is suppressed in order to allow the fetus to thrive. And other infections can make us more susceptible to numerous other infectious diseases. So until 2013 and the completion of the Human Microbiome Project, the uterus and its contents were thought to be sterile during embryonic and fetal development. And we discovered that that is not the case, that newborns are actually colonized in the uterus, and then that colonization continues during the birth process. So when a woman is pregnant, her normal vaginal flora changes dramatically, and certain organisms colonize the female vagina that don't normally colonize that location outside of pregnancy. And those organisms are really essential for the proper colonization of a newborn. Within 8 to 12 hours after delivery, a vaginally delivered baby has been colonized. Scientists also discovered that human milk is not sterile. Breast milk actually contains about 600 species of bacteria, as well as sugars that are not digestible by the newborn. So scientists believe that many of the sugars in breast milk are not there for the fetus, but they're actually there as food sources for these bacteria to ensure healthy colonization of the newborn. And that is the end of section 11.1. So here are checking in questions. Make sure you go through these and ensure that you can answer them correctly. Also, go back to the beginning of the presentation and review those learning outcomes. Make sure that you understand them. And our critical thinking question. The chart below illustrates the differences found in the normal biota of newborns born vaginally versus C-section. How might these differences influence future overall health? So this is a really interesting topic, actually. You can see in graphs A the difference between facultative organisms or facultative anaerobes and obligate anaerobes in a vaginally born newborn and a cesarean born newborn. And image B shows you the variety of organisms that colonize a vaginally born versus a cesarean born newborn, and the difference in their numbers. So compare the information here and then think about that question. How might these differences influence future overall health? Or would they? What do you think? Come back after you have sort of thought this out and constructed your response, and take a look at the answer slide and compare that to how you responded. And hopefully this video was helpful.